Let's unpack some of this biology a little bit to help people think about this better. So you mentioned this thing called the Furin Cleavage site. So can you explain briefly how this virus actually infects and gets inside of human cells, and what that cleavage site has to do with it?
So the virus's mode of attack is a sort of a sort of two-punch one. First, it latches onto its target, which is the protein called the ACE2 receptor. Then the second half of the virus's spike, the spikes that sort of jut out from the surface all over, has to be cut away from the first part so that it can then merge its membrane with that of the cells and inject its RNA into the cell and hijack the cell's protein-making machinery. So to make that cut between the two parts of the virus, the virologists call them the S1 and S2 parts of the spike protein. To make the cut between S1 and S2, the virus relies on enzymes that are naturally present on its target cell. And the bat virus relies on various other sort of bat-type proteins. But virologists have known for some time that you can really soup up a virus and make it a super human pathogen if you provide it with a way of recognizing a protein very, very commonly expressed on human airway cells called furin. So furin is sitting there to cleave various proteins that are sort of circulating in the human media. But if you insert into your virus's spike protein gene the specific sequence of amino acids that is recognized by furin, then your virus is going to have a much easier task of infecting human airway cells, because there'll be furin proteins all around to help cleave its spike protein, cutting off the S1 part, allowing the S2 part to deploy. So lo and behold, when you look at the SARS-2 virus, the most distinctive feature of it that comes out to any virologist is that right at the S1, S2 junction, just where it needs to have one, is a furin cleavage site. That's a sequence of amino acids that is recognized by furin. And the other remarkable feature is that SARS-2 is the only member of its viral family that has this site. Its viral family is called the sarbecovirus, and the SARS-1 virus and the MERS virus belong to it, and many other SARS-type viruses that the Wuhan researchers have found. So this is sort of quite a large family, and SARS-2 is the only one so far known that has a furin cleavage site.
So if you look at all of the sort of coronaviruses related to SARS-2, SARS-2 is the only one that has any trace of this site. And the site is basically the perfect sequence one would need if you were a virus who wanted to be infecting human cells, because you're taking advantage of a human protein that's actually necessary to clip that spike protein in order for it to get inside.
Yes, that's right. I should just say that the coronavirus is the sort of great big family of viruses, and the sarbecoviruses are a little subfamily within the coronaviruses. So there are furin cleavage sites in other coronaviruses. It's just that as far as the sarbecoviruses are concerned, it's only SARS-2 that has the furin cleavage site.
Mm-hmm. So is it fair to say that it's conceivable that such a site could have gotten right at that particular spot for this virus through natural evolution, but it would have been quite a remarkable mutation or a recombination event that would have happened to get it right there. But with the lab leak sort of hypothesis view of this, it's quite trivial because molecular biologists can just use standard lab techniques to put it right there.
Yes, that's right. On the natural emergence thesis, and the only way a virus could acquire a sort of a long string of new genetic material like this would not be through mutation, which does just sort of one base at a time, but it will be through recombination. That's when two very similar viruses infect the same cell, and they reassemble their parts, each with parts that belong to the other virus. So that's how viruses acquire new genetic properties. Recombination happens all the time. But the one thing you cannot acquire through recombination is a genetic element that your family of viruses does not possess. So since the sarbecoviruses, so far as we know, do not possess the furin cleavage site, it's hard to see how SARS-CoV-2 could have got it that way. It's much easier, as you say, to suppose that it was inserted there in the lab by people who wanted to soup up the virus. And now that we have this grant proposal to DARPA from the EcoHealth Alliance, we know that this is exactly the procedure that the Wuhan researchers had in mind.
So, DARPA is going to approve funding, approve grants that go to this thing called the EcoHealth Alliance, run by Peter Daszak. And then you're saying that group can then sort of subcontract or send portions of those grants out to places like the Wuhan Institute of Virology. How does that chain of command work exactly?
Well, these are US government grants. So the principal ones go from NIAID to the EcoHealth Alliance. And the other investigator, that's the guy who's sort of responsible for the research and the grant, is Peter Daszak. And he then subcontracts people at the Wuhan Institute of Virology, particularly Shi Zhengli, who is the chief Chinese coronavirus expert. Now,
so why is there? So EcoHealth Alliance, I understand that piece. EcoHealth Alliance is based in New York City. Is Peter Daszak himself running a lab in New York, and only a subset of the research goes out to places like Wuhan Institute of Virology, or how does that work?
Well, it's a very natural question because my having said he's a principal investigator, you assume he's a scientist with a lab, but he's not. He doesn't have a lab. He's just a grant raiser and administrator. He's a sort of research entrepreneur. So all the scientific work is done. I should say almost all the scientific work is done at the Wuhan Institute of Virology. And just to jump back a second to your question about the grants, the main grant from NIAID via EcoHealth to the Wuhan Institute it was a 5-year grant, and it lasted from 2014 to 2019. There was then a second grant from NIAID, which was quickly rescinded after the epidemic broke out. Now, the proposal to DARPA was made in 2018, and DARPA did not fund it. It was a bit too risky even for DARPA, but its importance is firstly in showing what experiments were being contemplated at WIAV. And it doesn't necessarily follow that the experiments were not done just because they weren't funded, because it's routine in research to use your money at the end of one grant to perform experiments that will support application for a second grant. So these experiments may well have been done even though DARPA did not fund the grant.
Yeah, that makes sense. So Peter Daszak is not actually running a lab himself. He's sort of a professional grant writer. Is this all virology research that the EcoHealth Alliance is funding? Is he a virologist? Is that his specialty?
It is virological research, but he's not a virologist. He has a Ph.D. in parasitology from the University of East London.
Interesting. So his job is basically a professional grant writer to fund virology research, but he's not a virologist himself.
Yes, that's correct.
I wonder how he got that job.